Okay. Well, hi, everybody. Happy Wednesday and welcome to Translate, a webinar about translational research in radiology. Today, we are fortunate to have Peter Caravan, PhD, and Sydney Montesi, MD, join us to talk about the potential of molecular imaging of pulmonary fibrosis for managing patients with interstitial lung disease and other clinical applications. Dr. Caravan is a professor in the MGH Martinos Center for Biomedical Imaging and co-director of the Institute for Innovation in Imaging, also at Mass General. In the Martinos Center, he leads a multidisciplinary and translational molecular imaging lab focused on the invention of novel molecular probes and their broad applications in cardiovascular, pulmonary, renal, and hepatic diseases, as well as in cancers. He received a PhD in inorganic chemistry from the University of British Columbia. Dr. Montesi is a physician in the Division of Pulmonary and Critical Care Medicine at Massachusetts General Hospital. She received her medical degree from the University of Rochester School of Medicine and Dentistry. She then completed her residency in internal medicine at Mass General and her fellowship in pulmonary and critical care medicine at the Harvard Combined Program. At Mass General, she specializes in the care of patients with interstitial lung diseases, and she also conducts translational and clinical research in the area of pulmonary fibrosis. Just a quick note about the structure of today's webinar. We'll have roughly a half hour of discussion, followed by questions from the audience. If you have any questions at any point during the discussion, please feel free to drop them in the Q&A box in Zoom. So I'd like to start by asking you a question, Sydney. Can you describe what are some of the major challenges associated with managing patients with interstitial lung diseases, and how have clinicians addressed these historically?
Sure. Yeah, thank you, Gary, for having me. It's nice to be here. So historically, treating patients with pulmonary fibrosis has been a particular challenge, namely because we haven't had any medications up until the past several years that have shown any efficacy. The primary problem in patients with pulmonary fibrosis is the lungs become progressively scarred. And as one can imagine, if your lungs are scarred, it creates great difficulty with your breathing in terms of oxygenation. And so as a whole, sadly, most of the patients with pulmonary fibrosis experience progressive disease. But one of the challenges in caring for these patients is that even as a whole, the entity is thought to be or known to be one that's progressive, of ongoing deposition of collagen and progression over time. There can be a lot of variability in how an individual patient does. So there might be some period of time in which an individual patient with pulmonary fibrosis has a period in which they're feeling well and their lung disease is thought to be relatively stable. And then for reasons that aren't entirely well understood, that particular patient can have a quick worsening of their disease. And this makes it really difficult in terms of caring for the patient because our current ways to assess how a patient is doing, aside from asking them what their symptoms are, really involve around CT scan of the chest, as well as measurements of pulmonary function, which are conducted by breathing tests. And these tests are very helpful in terms of diagnosis and monitoring over time, but it's really hard to do is for any one measurement for these tests to really say how the patient's going to do over the next 12 months. And another area of difficulty is that although we are fortunate now to have few medications that have shown some effect in treating these patients, the medications really have been shown to only slow down the progression of the lung disease. And you can imagine if you have an entity that's known to be progressive and it has a very clinical course, it's really difficult to know, how well your medications are working. And so in caring for these patients, I feel like the two main clinical problems is one, is their disease active or not? And two, are they responding to treatment or not? And then I've had the good fortune of getting to work with Peter over the past several years because my interest in caring for these patients with pulmonary fibrosis is paired quite nicely with the work that he's done and trying to be able to understand on a molecular level what's going on in the lungs and see if that can give us kind of a window in using kind of molecular imaging to be able to determine, how active someone's disease is. And then also maybe get a window in terms of, can you detect early response to treatment? And so that's kind of the landscape of what we're coming to when we think about how we might apply some of advanced imaging technologies, and really trying to see what are the areas of clinical need, and how can we help that.
You're muted, Gary.
Thank you so much. Can you give us a little bit of background about when you came to this problem, sort of how you identified it as a possible application of the work you were doing?
This is for me. Okay. Yeah. So in fellowship, we had the opportunity to do some research time, and I had the good fortune of working in the laboratory of Andrew Tager, who sadly passed away a few years ago. But he was very interested in the biologic processes or molecular processes driving pulmonary fibrosis, and coming to research with more of a clinical slant that and kind of meeting Peter and some other investigators in the Martinos Center, I became interested in this idea of is there a way to kind of do something in between of what's being done on the bench in terms of molecular pathways, but how can we do this in humans? This was particularly appealing to me because I did not have particular interest in doing animal work. And the idea of kind of doing things in humans and trying to kind of mirror what we're seeing in terms of clinical course, and could we tie that to something a little bit more molecular? And so that's how I got interested, and then that led to a series of grants and applications and some projects, and namely probably what we'll talk a lot today is using Peter's probe that recognizes collagen to see how we can try to answer some of these unmet needs in pulmonary fibrosis.
Great. So Peter, can you set the scene for us a little bit about sort of what you were doing at the time and your work leading up to meeting Sydney?
Sure. So, one of the things in my lab that we try to focus on when we develop a new molecular probe is think about, to look at processes that occur across, maybe different organ systems or different diseases where we could have a broader impact if at all possible. And so, a big area of research for us is in fibroproliferative diseases. So as Sydney mentioned, in pulmonary fibrosis, essentially you're getting a scar tissue replacing normal lung. And over time, as the lung becomes filled with scar, it can't function properly, and that's what leads to decline. And that process is true in other organs as well. It's true in the liver, in a lot of chronic liver diseases. It's true in the heart, and in diseases like atrial fibrillation, or in types of heart failure. Again, the heart muscle is being replaced by scar. In chronic kidney disease as well. There's a whole range of diseases, and so much so that about, I think, half the diseases that kill us have some sort of fibroproliferative component to it. And so we were interested in ways to measure that, because there are a number of different medical problems depending on, again, the organ system and the disease type. Do we want, is the need really to see disease earlier than what we can see it now? Is the presentation coming too late? Or in other cases, is the need one of prognosis? Can we predict better the outcome, which really plays into what we've been doing with Sydney. A third is, can we follow the effect of treatment or disease progression over time? Can we monitor things? And often in a lot of the cases, the tests that we have are just either not there, or they're imperfect, or they're somewhat invasive. Like you can think of, doing a needle biopsy, which carries some risk. And so we've been working on different types of technologies to quantify tissue fibrosis, and we've been working in the liver at first, and still are. And then I had the good opportunity to meet with Dr. Tager, who Sydney mentioned, who was really an outstanding collaborator. And so that's and he'd learned about our work in the liver and said, would this work in the lung? And so we, it was a great opportunity that one gets in working at MGH, right, where you're surrounded by these really talented physician scientists, and so we started working together, and sure enough, the technology does work in the lung, and so we went on from there.
Okay, right. And so you mentioned, I mean, with sort of the wide, variety and sort of diverse group of people at Mass General, people often come at problems from different perspectives, which is very helpful. But I'm curious if each of you could describe sort of your most important goals in developing and applying the probe, and also some of the significant challenges, from your respective perspectives.
Yeah, I can go first, perhaps. Well, I'd say that the main challenge over the past year and a half has been recruitment in the setting of COVID. So we've, with Peter's assistance, as well as several people on this call, Ravi and Ciprian, we've been able to translate one of Peter's probes, probe that is sensitive to binding collagen, we think more recently bound collagen, into humans. And that first was done in healthy volunteers, and then in patients with idiopathic pulmonary fibrosis. And you can imagine these patients have compromised pulmonary function. So with the kind of COVID, it's been somewhat challenging in terms of recruiting, especially given the fact that things shut down for a while. But we've made great progress to date, and recently have expanded from imaging patients with a certain type of pulmonary fibrosis called idiopathic pulmonary fibrosis, which is kind of the most common and probably the most well delineated in terms of kind of clinical studies and clinical trials. But we've really recently expanded to patients with pulmonary fibrosis with other entities, because it's becoming more and more recognized that pulmonary fibrosis itself can have this progressive phenotype, and identifying that is important. And the kind of the processes occurring in idiopathic pulmonary fibrosis are in many ways similar to pulmonary fibrosis in other entities. So we've recently expanded to patients with an autoimmune condition called systemic sclerosis. Where we would like to see, or where I would like to see this go forward, is really kind of thinking ahead in terms of not only can we predict how use this information to really predict how patients are going to do over time, but taking that a step further in a way in terms of seeing how this could be incorporated into clinical trials. So we desperately need new therapies. And as I mentioned, it's really almost impossible outside of a clinical trial setting when you have a placebo arm to really determine if a drug is effective, and thinking about ways to come up with early determinants of treatment response. And if you think about the end stage of fibrosis really being collagen deposition, or increase in the extracellular matrix, perhaps a collagen probe has high potential to be a surrogate endpoint. So within the next year, we're going to be starting a clinical study of an investigational therapy for idiopathic pulmonary fibrosis. And while the study itself will be focused more on safety, not a primary endpoint, but one of the main secondary endpoints is really going to be looking to see if Peter's collagen probe is sensitive to an early to detect early treatment response. And if that's the case, that opens up a lot of doors and avenues. And then other ways thinking about this is that oftentimes when patients come to see us, they have very, very well-established disease. And that becomes hard to treat in a sense that if we can only slow the disease down when people are already symptomatic, it's difficult what we can offer them. But thinking ahead in terms of, can we wind back the clock, or not so much wind back the clock, but can we use this technology to identify patients earlier? And so we're learning more and more about, patients that are at highest risk of developing progressive pulmonary fibrosis, more recognition about entities like, family members with pulmonary fibrosis, and kind of how could we use Peter's technology to really try to do early identification of people with active disease, and then try to think about how we can institute treatment early. So I think those are kind of two areas that I'm thinking about. And on those lines, I mentioned we have a study that's going to be coming within the next year using this technology and treatment response. And then we have another study we're going to be doing with collaborators over at Brigham and Women's, trying to investigate the utility of this probe and kind of early forms of pulmonary fibrosis. And as Peter mentioned, one of the exciting parts of this technology is that collagen is no way specific to IPF, and we can apply it to multiple other pulmonary diseases, and then thinking beyond pulmonary diseases. So people that are on this call that are applying this technology to pancreatic cancer, thinking ahead of like inflammatory bowel disease, and Peter and I actually have a collaborator in South Africa that's recently applied this technology to looking at fibrosis in active tuberculosis. So I think we have still a lot of exciting areas and opportunities to collaborate with the many talented people here.
Excellent. Peter, can you talk about some of the challenges you may have encountered in translating the technology to this point?
Sure. There's a lot of challenges. Sort of thinking back on the pain. So yeah, there's really a lot that goes into taking. This was a molecule that was invented here in Charlestown, and then went from chemists making the molecule and testing it in biochemical assays, to then working, like as I mentioned with Andy Tager's lab, and in different sorts of animal models, and showing that it worked in these animal models. So that and then this was work that was done by a really talented postdoctoral fellow at the time, Pauline Desaugiers, and she worked to show that this worked, and of course, the first compound that you make doesn't necessarily work that well, and there was a series of optimization, and then going forward, we've had something that worked well in mice. And then we wanted to make sure, though, that the target and what the signal that we were seeing in mice we would also likely see in humans. And so, Pauline would get up at 4 a.m. when there was a phone call, when they were doing a lung transplant at the hospital, and she would wait outside the OR, and they would come out, and working with a great pathologist, Lita Hariri, would get identify certain parts of the diseased lung, come back to Charlestown, and then test to see whether her probe, recognized the diseased lung in preference to healthier lung. So all of that came together, and then so then the next steps were really doing some safety testing and manufacturing development with the PET center downstairs with Jacob Hooker's group, to now take this forward into humans and put together a big dossier for the FDA to approve the use of this novel imaging probe in humans. And so that ended up also having some issues, if Sydney can recall. But we overcame those things that were really out of our control, like supply chain issues in one case. In another case, we were doing a toxicology study with an external vendor who then screwed things up and had to redo the whole study again, and that set us back a little bit. But finally, we persisted and we came through. And even then, we started out and we were getting great results. And then the state and the national the regulatory body that regulates radiation use changed some specifications, and then we had to switch from our source of isotope, and we had to start all over again with manufacturing, and that set us back. But we, but through the whole process, I feel like, you just can't give up. And so, so we've had, really a great group of individuals who's been, very, very persistent. And so I think, that's, and that's what's really allowed us to keep moving forward. And, and so the work with Sydney, really started this, and working in idiopathic pulmonary fibrosis, and the findings there have been quite positive. And as she mentioned, that work is broadening out, and then we're broadening out further with, as other people learn about this, others want to use the probe for other indications, like another colleague, Michael Lanuti, and is a thoracic surgeon, and he's also using the probe to look at the effect of radiation injury in the lungs. So he sees cancer patients, and one of the side effects of radiation therapy is the possibility of actually making the lung fibrotic. And so, could we identify that earlier? And then if we could, maybe we could help prevent that in those patients. And as Sydney mentioned, working now, she's got collaborations with a group at the Brigham, and then with this sort of multi-center study looking at treatment response. So it's really nice to see the technology grow. And one of the things that we're working towards in our lab as well is in really starting to disseminate this out to other centers.
As you disseminate out to other centers and sort of broaden the applications, do you find you need to tweak the technology, and then does that sort of put you back in that sort of iterative process of developing it?
Well, yeah, so this is where it's very different from hardware, where if you think about the work we do at the center, and improving, MRI technology or PET technology, it's much harder to make improvements, because if you make an improvement to the imaging probe, you've made a new chemical entity, a new molecule. And then the FDA says, no, you've started over again. You have to go right back from zero and show us that this is safe and effective. And so they're really, in order to make a next generation probe, there really needs to be a good, compelling reason, I would say, to kind of go back to that process. Once you've gotten here, I think we wanted to really take this as far as we can find it. At the same time, this probe is teaching us new insights about human biology, and then prompting us to explore, other aspects of that biology. So we are developing other probes that will be, I would say, complementary to this one.
Okay, great. Very good. Yeah. Well, so what is next then? Sydney, you've mentioned a number of different sort of studies moving forward and applications. Are there any that you are particularly excited about, or most excited about?
Well, I myself am most excited about whether or not we can detect early treatment response, because for pulmonary fibrosis clinical trials, the duration has been 12 months. And that's a long time, especially because patients can have a lot of disease progression during that time. And as Peter has said before, kind of time is long, especially if you're not correct in terms of your treatment. And it's not so much now being not correct, it's just we need more therapy. So I think that I'm particularly interested in that of the different applications on the pulmonary side. I think that has garnered probably the most enthusiasm, and so to speak, and especially in an entity like pulmonary fibrosis that hinges so much on imaging in terms of diagnosis and monitoring. This is a technology that pairs nicely, and talking about kind of obstacles or things to think about when translating for other centers is the fact that our imaging to date has been with MR, but most of pulmonary medicine is kind of CT based in terms of using that as a diagnostic. So things that we're working on with Ciprian and other people in his lab and Peter's lab is thinking about, kind of ways to, in terms of thinking about the analysis that's relevant from what we're doing on PET-MR, but also thinking ahead, because not every center is going to have a PET-MR machine, and it's going to be PET-CT. So that institutes a whole other host of complexities, as well as exciting opportunities in terms of, attenuation, correction, et cetera, and things that I'm learning about, and defer to Ciprian for his expertise. But I think there are a lot of exciting things in the horizon, and hopefully within the next 2 years we might have some interesting preliminary data on our ability to detect early changes if this does work, and that would support data that Peter has in the animal models in terms of this probe being able to detect treatment response.
Great. Peter, do you have anything particular you're looking forward to on the probe development side?
Well, yeah, I think, one of the things that the idea of treatment response, both with established treatments and then also with new therapies that are coming along. Like if you look in the drug development space, there's an awful lot of activity around IPF new therapies, and unfortunately for the most case they haven't been successful, and there's a number of possible reasons why, but one of them goes back to this point that Sydney raised earlier, where patients with this disease, we know that they unfortunately from diagnosis they have poor prognosis over the period of, say, 4 to 5 years, but over a course of a year they could be remarkably stable. And so if you think about doing a clinical trial, then the people that are stable over the course of that period, then it's going to look like the drug is working, or if they're on placebo, that the placebo is working, right? And so what you would really like to be able to do is to identify those patients that are going to have a decline over time, where you could really see if the drug is having an effect or not. And that, I think, would be incredibly valuable, and it's really something that we're both really keen to try. And we've certainly had a lot of interest from academic colleagues that are testing new medicines, and also from our friends in industry as well, for this reason. I think this is really a burning need, and I think this is really an area that excites me in general. How can we reduce the failure rate in drug development? How can we get drugs to the market quicker? And I think that this sort of personalized imaging can really play a big role there, both in the lung and elsewhere.
Okay. Well, great. Well, very good. Well, we're right at about a half hour. So if anyone has any questions, please feel free to drop them into the Q&A box. Maybe we can talk a little bit as people are formulating their questions, sort of about the Mass General environment, and what do you think about this environment is really conducive to translational research in your experience? Sydney, start with you.
So I think I've benefited immensely from this environment, and I think one thing was helpful, having worked with Andy Tager. We were all in the Charlestown Navy Yard, and so it made for kind of easy conversations with Peter, and I think that so coming from the medicine division, I bring a little bit different perspective, but I think one thing that's been incredibly helpful to me is just how the Martinos Center has just been unbelievable in terms of kind of supporting clinical studies and trying to kind of giving me the kind of flexibility to make some of these things happen. And I think that these cross-discipline collaborations are specifically very helpful, both within different medicine subspecialties, thinking about applying this technology to other organs with fibrosis in terms of like across department. So I think that just kind of collaboration and is, without that, I don't think that's this project would be where it is.
Right. Excellent. Peter, do you have you had a similar experience?
Yeah, and I think I've had a number of really great experiences collaborating with Sydney and other physician scientists, and I think the sort of common theme throughout that is everybody that I work with who sees patients, it's really evident how much they care about their patients and how much they really want the best for their patients, and so they recognize and they are happy to help and adopt to try and adopt new technologies that could provide that edge, could provide that benefit. And so I think that's I think that's really a common thread throughout MGH, and certainly it's a hallmark, I think, of working with Sydney.
Mm-hmm. Excellent. So, well, we don't see any questions in the Q&A. If anyone has any, feel free to jump in. So we'll give them a few seconds to do so. Thank you both for joining us. Anything we haven't covered that you'd like to mention?
There's just one other thing that I think is worth bringing up, just about the concept of molecular imaging in general as applies to pulmonary fibrosis. In the past 10, 15, 20 years, I mean, the field of oncology, if we just take lung cancer, it's just exploded in terms of treatments and advances, and so much of that has really been able to do molecular phenotyping on tissue, and we don't have that in pulmonary fibrosis because while we do get biopsies, not every patient is able to undergo a biopsy for various reasons or comorbidities. And then a biopsy itself of the lung carries a insignificant risk of acute worsening of the lung disease. So thinking about how we can use probes like the ones Peter's developing, or maybe other probes that are being developed in the center or from collaborators across the country, to really try to get a better understanding and see if we can, within a disease that's known as IPF, are there multiple different subtypes that only through kind of imaging technologies with very specific probes, we might be able to identify, okay, this is IPF type A, this is IPF type B, and then type B might respond best to, medication C. So I think that I just bring this up because I know there are people in the center working on different molecular imaging probes or technology, but I think all this is really helpful in terms of clinical diseases where we don't have the opportunity to be so reliant on tissue.
Great. Very good.
Yeah, and I sort of echo that. I think that we're seeing certainly and again, also in the oncology space where imaging, molecular imaging is moving that way. There's been a couple of new products approved recently where you need a positive imaging test that's going to select the therapy that's going to follow, and applying this to the lung or to other pathologies is really quite an obvious thing, and it's also this is the severity of this disease is not to be understated. I think it's really it has a worse outcome than most cancers, and so really there's really an urgency around better ways to treat these patients, and if we can help identify which therapies would work better, if we can identify new therapies through imaging, I think this is this is incredibly valuable.
Excellent. Well, thank you both so much. I really appreciate your time and coming in and talking to us. Fascinating discussion. Well, thank you everyone for joining, and we will see you all soon.
Thanks, Gary.
Thank you.